So thank you all for coming, and it's an honor to introduce Justin Kumar. So I thought I'd start with a little bio first. So he got his bachelor's at University of California on Riverside, and then he got his PhD at Purdue with John Reddy. For those of you who know YI development, you will know John Reddy, and also then he moved to University of Southern California for his postdoc with Kevin Moses. And after that, he started his faculty job at Indiana University in 2002. And there he's moved up from assistant to associate to full professor. Couple of highlights: he's honors. He's a fellow of the American Association. What am I missing? I'm missing an A.
Advancement
of Science. Yes, AAS. He has also won campus-wide teaching awards at Indiana. And looking at the CV, it's clear. So from 1 year in as an assistant professor from 2003. Since then, he's been continuously funded by NIH, which is quite impressive. And you know, I met Justin about almost 20 years ago at a flying meeting, and I was following his papers before then, but I've certainly followed his papers since. And there are a couple things that really stand out. And to me, these are things I really admire. One is focus. So he has been focused on eye development, particularly Drosophila eye development, his entire academic career. By entire academic career, I mean since an undergrad. As I found out at dinner last night, it was week one of freshman year in college, where he started working in a lab, then worked in eye development. He had 3 papers as an undergrad, research papers, including one that actually had a significant contribution to understanding how you actually like the waves of development that happen in the eye disc. But there's a side story. Maybe you can come up with the graduate student talk. Like as a 18-year-old, he has access to cesium, like radioactive cesium. So that's interesting. But anyway, what I was getting to is the focus. So since that point, he's been focused on Drosophila eye development. The other thing that's really special with Justin is how beautifully he builds stories, right? And you will see that today in his talk. But you know, there are certain journals that are really, you know, close to my heart, like genetics. He has published 6 genetics papers. He has like 10 developmental biology papers. But to top it all, development, the journal of development, he has 16, maybe one more in the way.
Yes.
17 development papers. So the sort of storytelling, the quality is absolutely amazing. So with that, I'll let him tell us the story today. Thank you for coming. Welcome.
So thanks, Arash, for that very sweet introduction. The buildup was pretty high, so I hope that it's going to match it. So what I'm going to do is try to tell you a story today about how you can get 2 different organ fates by simply manipulating a very common gene regulatory network. Just changing its levels up and down can actually switch the fates between these 2 organs. And that's somewhat unusual because you normally think of 2 different organ fates of having 2 different gene regulatory networks with different combinations of transcription factors and signaling pathways. But we stumbled onto one where it's a common pathway that is used just at 2 different levels to give you 2 different things. So I hope by the end of this, you're convinced by that story. And it turns out that the developing eye is a really good place to look at this. I can somehow move. Right. I think we should work. Yeah, perfect. Okay. The developing eye is a great place to study this because the eye really differs across animals throughout the animal kingdom. We have lots of organisms that have variations on camera eyes, like you and me. We also have variations on compound eyes, which are seen in insects. And there's quite a bit of variety between those two things. And all of these are thought to have arisen from a prototype eye that Charles Darwin spent a lot of time talking about in The Origin of Species. And he imagined that the original primitive eye consisted of a single photoreceptor neuron, a single pigment cell, and a translucent skin cell. This wouldn't give you any resolving power to know what you're seeing, but you could certainly sense light, you can certainly sense shadows. And if that photoreceptor was connected to a set of ciliated cells, it allowed that primitive organism to actually move and get out of the way or move towards something. So within that context, how do we get all of these different eye types? And there's a couple of different ways that people have thought about it over the years. One way that people thought about is that the eye actually has developed at least 40 individual times and maybe 63 different times through evolutionary history. It was a famous paper published in 1977 by these two fellows that really proposed this model. And the idea would be that once you go from that primitive photoreceptor, you would actually have different gene regulatory networks that would get rewired and incorporated, and you can get all the different possible eye types from these variations on the compound eye to these variations of the camera eye. And some of the evidence that was used to support this came from the class bivalva, where you would see organisms like the hard-shell clam that would have a camera eye, very within the same family as ones that would have different types of compound eyes, these ones lacking lenses. So the idea was that they proposed that you could get these sort of individual evolutionary steps within very closely related species. However, the counterpoint to this came from a set of remarkable studies in the 1990s in which a number of different individuals, including Walter Gehring, had noticed that mutations within the PAX6 transcription factor were the underlying cause of eye defects from within Drosophila, zebrafish, mice, and humans. And in the case of humans, the disease that we're looking at here is aniridio, so being born without an iris. And it was Walter who had actually proposed that the eye actually has a sort of monophyletic origin. So the idea would be that you would go from a primitive photoreceptor you would develop a gene regulatory network that was in common that would give you that prototype eye that Charles Darwin had mentioned. And from that point onwards, you would get sort of fine scale gradation phenotypes or changes that would give you all of these different eye types. And so one idea that we had in the lab was whether you could take a common gene regulatory network, and as I mentioned in the introduction, sort of change its levels and switch from one different eye type to another. And we're not going to do anything as ridiculous as going from a compound eye to our camera eyes, but we're going to focus on two visual systems that are within the Drosophila head. Now, some of these images, when they project, seem a little bit darker than my computer. So if there's something that I'm pointing to that you don't actually see, please raise your hand and tell me so that I can point them out to you. So what you're seeing here are either standing electron micrographs or light microscope images of different portions of the fly head. And the two features that we're really going to talk about today are these compound eyes. There's two lateral ones shown here. They're made up of about 800 identical hexagonal units. And these three simple eyes that are on the dorsal vertex of the head, these are going to be three simple eyes. And what I'm going to show you a little bit later on is how that they're anatomically very different from each other, how they have very different physiologies, how they project their axons to different parts of the brain. And there was a recent paper in eLife that had suggested that the ocelli actually evolved first, and it was actually followed by the compound eyes much later in evolutionary history. One of the key sort of stars of our talk today is going to be the PAX6 transcription factors I introduced from Walter Gehring's work. And so PAX6 belongs to a large superfamily of transcription factors that are called the PAX family. They originally started as a protein that had a homeodomain The idea is that a TC1 transposon, which has a domain that looks very similar to a modern day paired domain, that transposon inserted into this coding gene and gave you a PAX precursor that now has two DNA binding motifs, a paired domain and a homeodomain. From that original precursor, there were multiple duplications over evolutionary history. And the one that we care about today is this molecule PAX6. And in particular, we care about two Drosophila PAX6 genes called Ilus and twinovilus, and we'll talk about them actually quite a bit. So I'm just going to bring up their structure for a moment and just remind you that there's two domains, a paired domain, a homeodomain, and there is a activation domain that's at the C terminal. It's a PST activation domain. This transcription factor can bind to DNA using many combinations of its DNA binding protein. So it can use the entire paired domain in combination with the homeodomain. It can use actually two individual subdomains, either individually or in combination with that homeodomain. So it has a plethora of different DNA binding targets that it can actually sit down on and regulate. Now, as Walter had shown, and I've shown you some images of how PAX6 is required in all seeing animals, it's actually part of a larger gene regulatory network that we refer to as the retinal determination network. So there are 14 transcription factors that are sort of woven into this interlocking system where each of these arrows represents a molecular input, either activating or inhibitory. And here are the lists of the fly genes, of which here's Ilus and twinovilus. There's two other molecules that will become really important. A downstream member Synoculus, which is a DNA binding protein here, and its cognate binding partner izapsin, which is a transcriptional coactivator. So most of the fly genes have at least one copy in vertebrates, and mutations in a fair number of these will either lead to defects in mouse model systems or patients with congenital retinal disorders. So the network seems to be, as a whole, conserved across evolutionary history. What makes this network kind of interesting is both its loss and gain of function phenotypes. So if you knock out any individual member of this network, you actually will have dramatic reductions in the compound eyes. You will either lose them completely or they'll be reduced pretty strongly. The more exciting feature, however, is what happens when you overexpress them. So again, you can take any one of these and overexpress them in non-ocular tissues like the legs, the wings, the antennas, the genitals of a fly, and you can actually redirect portions of that tissue into compound eyes that you can see here. They're all across the legs. The genitals out of focus, but there are compound eyes there as well. Those are anatomically identical to the endogenous compound eyes. They can actually respond to light with electroretinograms. If the compound eyes are actually located somewhere on the head, you can actually get a second order response from the brain. The ones that are farther away do not connect their axons to the brain, so you only get the initial response and not a second order neuron response. And to kind of give you a sense of sort of this evolutionary conservation, George Halder, a friend of mine and a friend of Arash's when he was a grad student in Walter's lab, had actually taken the mouse version of PAX6, which is called small eye, had driven it in Drosophila, and you can actually now redirect parts of the fly into developing compound eyes using the mouse gene. And you can do that with any one of these other network members. So they're both present, they're necessary, and they're evolutionarily sufficient to replace each other. And as you'll see later, these things are expressed both in the compound eyes and the ocelli themselves. So this entire network has been shown to directly regulate genes that would command every step of an organ formation, whether it's initially tissue specification, proliferation and growth, pattern formation, compartment boundaries, all the way down to axon guidance. So these things actually direct every step directly, and it's not just a sort of a general mechanism. So as I mentioned, what we're going to do is we're going to try to manipulate members of this pathway and see if we can get the compound eyes to transform into ocelli and vice versa, with the idea that using the same network in two different tissues at different levels may be a mechanism for giving you a diversity of tissue and cell fates. Okay, so what I'd like to do is kind of tell you about how these things are structurally different internally and what kind of features of behavior do they sort of control. So the compound eyes, each one has about 800 units. Each unit has an identical set of about 20 cells. There are 8 photoreceptors, a set of lens-secreting cells, and a set of optically insulating pigment cells. And you can see the photosensitive membranes of each photoreceptor here in these black dots. These are called the rhabdomeres. And what you can kind of tell is that each one of these units is an identical replica of its neighbors. The only difference is that the photoreceptors are pointed in mirror images to each other across this imaginary sort of midline down the middle. And these things also have what's called an open rhabdome formation, where each rhabdomere is actually pulled apart from its neighbors. In many insects, all the rhabdomeres project into the middle to form one large rhabdome, which is sort of a closed rhabdome formation. Now, if we think about what the compound eyes are responsible for, their main thing is to really detect motion. They can also detect ultraviolet light through some of the inner photoreceptors. They can also detect color vision. That's a lot of work that Claude Desplan has done. They detect polarized light, and they're huge for entrainment of their circadian clock. Now, in contrast, the ocelli, which are these 3 simple eyes at the top of the head, they have a single lens that will actually cover about 80 photoreceptors. And all of those pigment cells are not interspersed through the photoreceptors as you see in the compound eyes, but they actually ring sort of the surrounding areas. And when you look at their internal structure of the photoreceptors, you can see that a lot of these rhabdomeres actually interact from neighboring cells and they actually touch each other. It isn't necessarily one huge closed rhabdome with 80 rhabdomeres, but there is a lot of conjoining of rhabdomeres from adjacent photoreceptor cells. So they're structurally quite different than each of the individual ommatidia of the compound eye. And if we look at what they do, they're actually doing quite a lot of different things. So they're very minor entrainers of the circadian clock. They're actually responsible for fast response times. So when you're trying to swat a fly, it's the ocelli that are actually allowing them to escape, because the ocelli bypass many different layers in the brain and they go down to the central brain complexes, while the ommatidia actually go through the lamina and the medulla, and then second order neurons will go down to the lobular plate and so on. They also help for flight stabilization. So as flies are flying, the 3 ocelli that form this little triangle, as light strikes one, it'll cast a shadow on the other. The flies can sense that, communicate that information to the wings and the halteres, and readjust their flight. So rather than rolling or pitching to one side, they can actually sort of straighten out. And then based on their rhodopsin expression patterns, they can actually detect very dim light. So insects that fly below the canopy, like say in the Amazon, where light is 1,000,000 times dimmer, will use their ocelli to actually fly around. And then there are some ants, a few land-bearing ants that have ocelli. They will actually detect light from stars and use celestial navigation to sort of move around. So again, they have very different axonal projections, lots of different behaviors, and they're structurally very different from each other. And as that paper I mentioned suggested, that they evolved quite at different points in evolutionary time. So both of these 2 adult structures are derived from a larval tissue called the eye antennal imaginal disc. So all of the fly external features come from these sac-like structures, and the Drosophila head has 2 eye antennal discs. Each one of these discs will give rise to about half of the head. You get one of the 2 compound eyes. You get 1.5 of those 3 ocelli. You also get the 2 olfactory organs, the antenna and the maxillary palps. And then all of this head epidermis that surrounds these organs come from these 2 eye antennal discs, sort of the surrounding cells around the edges of the disc. Now, this disc arises during embryogenesis, where you have cells from about 6 different embryonic head segments will actually sublime themselves together, forming one entire structure. As that embryo hatches into a larva, you get sort of this sort of teardrop-like structure that forms that eye antennal disc. If you look at gene expression profiles, basically they're uniform through the entire tissue, as I'll show you in a few slides. As you go through the first, second, and third larval instars, gene expression profiles will change, as I'll show you, but also the shape of the disc changes, where now you can begin to see the oval that will give rise to the compound eyes over here on the left, and the circular structure that will give rise to the antenna and maxillary-palps over there on the right. So that tissue, as I mentioned, is a sac-like structure. It has two layers. It has the disc proper layer that lays underneath. That's where all of these tissues are really being specified. There is an overlying set of tissues called the parapoidal epithelium, and these things are kind of zipped together so that you get a small lumen that comes in between those two major layers. The cells around the edges are called the margin, and it's basically like a closed pillowcase. And what you have is this, creating this lumen allows for signaling molecules from one cell to not like just basically diffuse away into the hemolymph. It actually has this little space that contains it. So cells over here can signal molecules to neighboring cells. We have signaling pathways that are expressed in the peripoidal epithelium that will secrete molecules down and towards the disc proper, and vice versa. So the beginning of the third larval instar is where you have pattern formation in the eye disc. So at the posterior margin, a wave of differentiation will initiate. The anterior edge of this is called the morphogenic furrow, and that furrow will actually migrate from the posterior end all the way to the border between the antenna and the eye, the most anterior margin. And as it does this, the sort of sea of undifferentiated cells will be transformed into columns of unit eyes or clusters of photoreceptors. So those adult columns that you were able to see are actually being formed asynchronously throughout the disc. Now, at this stage in development, where I've shown you these pictures, the ocelli have not yet to form. So they form a little bit later, and that's kind of shown to you in the next slide, where we're looking at two stages of eye antennal discs that are staying now with an antibody against the izapsin protein. That was one member of that retinal determination network. And you can see that at around a stage that was similar to what you've seen on the previous slides, you can see that there are antibody or izapsin is expressed in cells that are still beginning to differentiate, deciding to become unit eyes. You can see that izapsin expressed in all of the differentiating photoreceptors behind the furrow. And now a little bit later, you can see these 1.5 ocelli actually showing up. And so these guys actually turn on a little later in development. So what I'm going to do is kind of show you a slide next that has a number of different mutants on there, just to kind of orient you with what we're going to look at through much of the talk. So you've seen this wild-type image on the top left, where here are the 3 ocelli. In 1926, this guy named Tom Patterson had identified a mutant that he called ocellilus because it eliminated the ocelli and these little bristles that were found in between. Now, members of the retinal determination network will also lose the ocelli. So you can see here this gene cynoculus that I had introduced to you earlier. It's expressed both in the compound eyes and the ocelli, and you can actually see that when in this particular mutant, which removes a visual organ-specific enhancer, that you lose the compound eyes and you lose the ocelli itself. In the case of izapsin, we have deletions of enhancer elements that get rid of the compound eyes, but we know it's expressed in the ocelli, which I had just shown you. So what we had done is we had taken a RNAi line that can be driven by a GAL4 driver that's expressed just within that ocellar region in the disc, and you can see that the 3 ocelli are actually missing. So the retinal determination network as a whole is required both in the compound eyes and the ocelli, and that's where our story will kind of begin. And this was just to remind me to remind you that we're going to look at two different PAC6 molecules, Ilus and twin of Ilus. And so there's a lot of evidence to suggest, and some of it I'll show you, is that Ilus and twin of Ilus function within the developing compound eye to specify its fate, but that only twin of Ilus gets used in the ocelli. And there was lots of early evidence to suggest that these two PAC6 molecules were binding to two different sets of target genes, twin of Ilus giving you ocellar specific gene expression, and Ilus giving you compound eye specific gene regulatory networks. When we look at twin of Ilus mutants, we can see that we made a CRISPR deletion of the entire TOY1 locus And you can see that 2 out of the 3 ocelli have been removed. If you knock it down with the toy RNAi line, you can see in this case, all 3 ocelli are missing. So between these RNAi knockdowns and loss of function mutants, there's a variable number of ocelli that will be missing at any one time, suggesting that there are additional inputs into that field outside of twinovirus. In contrast, we have a number of mutants of ILUS, one of them being a null allele in which you have a 9 kb deletion that takes out the entire transcriptional start site and there's no transcript. There are a number of transposon element insertions into an I-specific enhancer. We also used CRISPR in a previous paper to delete that enhancer completely. In all cases, you have these compound I defects, which you've seen before, where you have this either complete loss or a severe reduction. In all cases, these mutants drop ILUS transcript levels, as shown by qPCR, to less than 2% of wild type levels. So these are all strong alleles, but none of them seem to affect the ocelli. You can see in every single case, you might affect the compound I's to varying degrees. Here's the null mutant where the compound I's are completely missing. But in all cases, whether these are the 6 mutants that we have or an RNAi line, you can see that the ocelli they're present and they're robust. So it does suggest, at least genetically, that that ILUS is not involved in ocellular development. It's only twinovirus. And this is where our starting point or jump off point was to try to investigate this process. So what we had done is we had actually looked at the expression patterns of these 2 transcription factors beginning at the middle of the second larval instar. So here are antibodies looking at twinovirus in the top row and ILUS in the bottom row. If you look at twinovirus first, what you kind of notice is that, as I mentioned, gene expression is throughout the entire I-antennal disc really early. But as development proceeds, you start to retract twinovirus expression from the developing antennal field. However, you do have it maintained within the area that will give rise to the ocelli. And that's consistent with when you knock out twinovirus, you lose the ocelli in those mutants that I showed you. In contrast, if you look at ILUS, it does start off uniformly. It gets retracted as twinovirus does, but you can see that it starts to actually retract from that ocellular domain as well, so that by the end of the third larval instar, there's a clear zone where ILUS protein is no longer there, and this is where the 2 ocelli will develop, but twinovirus remains. So we were interested to know kind of why is ILUS on early, and why does it actually get retracted during development? Is it because ILUS and twinovirus are acting 2 different gene regulatory networks, so you want to pull ILUS away so that you can promote ocellular development with twinovirus? Or, as the model that I'm trying to propose to you, are they actually binding to the same set of targets, but you don't want those targets to be turned on at too high levels within that ocellular domain, so you're retracting one of those PAX6 transcription factors, so you can maintain a lower level of expression that would give rise to a totally different visual system. So those are the 2 models that we're trying to look at. So in terms of the evidence as to why we think that ILUS might be binding to the same sites as twinovirus, are they regulating the same targets? It comes from this mutant here. So this is an odd ILUS mutant. It's a dominant negative allele. So as a homozygous viable mutant, the thing is dead. But as a heterozygote, you have these severe reductions in the compound I's, which is similar to the homozygous loss of function mutants. But what's interesting is you can see that the 3 ocelli are completely missing. But I just told you that the ILUS loss of function mutants actually have no phenotype. And during ocellular development, when twinovirus is specifying its fate, ILUS is completely retracted. So why do you get this no ocellular phenotype? And it has something to do with the type of mutant that we're actually looking at. So it turns out that this is a truncation mutant that kills one of the 2 DNA binding motifs, and it gets rid of the activation domain. So all that's left is the pair domain. And there's lots of transcription factors that, when mutated in this way, behave as dominant negatives. And the idea is that this mutant protein would be binding to its targets through the pair domain and preventing the wild type version of ILUS to actually bind. But I just kind of told you that Islus doesn't play a role in this at all. And so it suggested that this mutant protein was actually interfering with another transcription factor's binding sites. And the most obvious candidate would have been twin of Islus. If we look at how Islus and twin of Islus function in the compound eye to sort of further this idea that they may be acting redundantly, we looked at the two null mutants of the two genes. And what you can see is that twin of Islus null mutants have pretty good compound eyes. They make almost as many ommatidia as the wild type does. You can see that qualitatively and quantitatively. The Islus null mutant, even though it has about half the number of wild type or the twin of Islus mutant, it actually makes a reasonably decent compound eye. And if you look at all the different cell types, all of them are present. And so they seem to be functioning redundant to each other. We just finished a round of cut and run where we're looking at the binding profiles of these two transcription factors. And it looks like that they're binding to the same places within the genome. I just didn't bring the data with me for this talk. So they are binding to the same places. So one question is, well, okay, if they're binding to the same places, we want to retract. Why do we want to retract one of them away from the developing ocelli? And it has something to do with the difference in the severity of these two phenotypes. If they're binding to the same targets, they're expressed at relatively the same levels, which I forgot to mention. Why do you have one that is much more severe when lost compared to the other? And that's from the data comes from a paper that we published about 15 years ago, in which a research associate in my lab, Bonnie Wiesner, had not only identified the PST activation domain within the C-terminals, she was able to show us that Islus was a much stronger transcriptional activator than twin of Islus. So how do we do that? So her first inclination was in this LAC-Z assay, where we had a UAS LAC-Z reporter in yeast. We had actually fused the wild type versions of either Islus or twin of Islus to the DNA binding portion of Gal4. So that chimeric molecule can sit down on this reporter and actually activate it and turn your yeast cells blue. When you look at the full-length versions of these molecules, you can see that Islus turns it on at a much higher level than twin of Islus. Qualitatively, it's a darker blue than it is over with twin of Islus. But these kinds of colorimetric assays are notoriously finicky and unreliable. So we actually switched to an assay where we can actually quantitate its strength. So in this case, instead of activating a LAC-Z molecule, we're actually going to try to activate a US His3 enzyme in a strain of yeast that are lacking the His3 gene, and we're growing them in media that lacks histidine. So there's no way for that animal or that yeast cell to make histidine unless you can activate this enzyme and get the pathway to actually work. So if you take twin of Islus and Islus, fuse them to the Gal4 DNA binding domain, try to transfect them into these cells, activate this reporter, you can now rescue and get those cells to survive. But now if you grow those cells in different levels of an inhibitor, 3AT, which actually inhibits the histidine biosynthetic pathway, in order to get these cells to survive in increasing concentrations of the inhibitor, you actually need to turn on your reporter at much higher levels. So what you can see here is that as soon as you raise the concentrations of the inhibitor past 35 millimolar, twin of Islus fails. It can't activate it strong enough, and you no longer have any cells survive. However, twin of Islus can do it all the way up to about 150 millimolar concentrations, suggesting it is about 5 times stronger in activation strength. So as we're kind of building through this model, the idea is that Islus and twin of Islus bind to the same targets in both organs. One is a very strong activator and one's a weaker activator. You want to have both of these in the compound eye to turn on your targets to a level to give you a compound eye fate. However, in the ocelli, you want to tune down the activation of that network, so you repress the expression of the strong activator and you leave only the weak activator on. So do the strengths of these activation domains actually have are they biologically relevant? I've shown you the loss of function mutants, but that could be due to different parts of the protein. We're going to try to tie this to the activation domains. So I'm going to switch gears for one moment, just a detour to show you that those C-terminal domains are actually biologically relevant. So I told you that both molecules can induce ectopic eyes, but they do so in very different patterns and in efficiencies. So in this assay, if you overexpress iris, you can induce ectopic eyes in a wide range of tissues in the body. Twin of iris, not so much. And I made a diagram to make this a little easier to see, where iris can drive ectopic eyes within the antenna, the wings, the flight balancer halteres, and in multiple locations within the legs. Twin of iris, on the other hand, does this very anemically. It only can turn on a single or give you a single ectopic eye within each of the different legs. And we can actually control where we put these things in the genome, so we're expressing them at the same levels. We've tested it with both mRNA, qPCR, we looked at protein levels. We're getting same levels expression of these two factors. So these things seem to be really different outputs when you drive these two things. So we then made a series of chimeric molecules in which we replace either the pair domain, the homeodomain, or the two C terminals, the strong and weak activation domain, and asked, well, which ones give you a strong ectopic eye pattern and which ones give you an anemic pattern? And so all the molecules here on the left, the one thing that's in common is that they all have the strong activation domain from iris, and they all give you this very robust pattern of ectopic eyes. Everything on the right gives you this very anemic pattern of ectopic eyes, and the one thing in common is that they all have the weaker activation domain from twin of iris. So it's the strength of the activation domains that seems to determine whether you're going to turn on networks at high or lower levels. And we're arguing that you want to retract the stronger one because you want the downstream targets to be turned on at a lower level. So if that's true, we should be able to look at the expression patterns of the downstream targets and see that they're differentially activated in the two visual organs. So I'm going to show you 3 direct targets on the following slide. So we're going to look at cynoculus, which is a direct target, izapsin, which is a direct target, and doxin, which is a direct target. There should be arrows from these two that bypass the doxin, which I forgot to draw on here. So what we did is you can see their expression patterns. They're in the compound eyes in various patterns, and then you can see they're in the ocelli to varying degrees. What we can do is take signal intensity from these expression patterns within the disc itself. We measure 4 different locations, differentiating photoreceptors, undifferentiate cells in the compound eye, and then the 2 ocelli. We measure their signal intensity over multiple discs, and then we average the results. And what you can see for both cynoculus and doxin on the left and right, you can clearly see that the expression patterns of these 2 factors in the ocelli are much lower than they are in the 2 regions of the compound eyes. Izapsin, on the other hand, seems to be expressed equally bright in both places. So what we wanted to do is take that differential expression and ask, what happens if you overexpress either of the 2 PAX6 genes? What happens if you overexpress either 2 of these targets, cynoculus and doxin? In the ocelli, if you raise their levels to equate to the compound eyes, do you get a change in their fate? To do this, we've been using this DVE GAL4 driver, which was listed on past slides. You can see here it's expressed just within that domain that will give rise to the ocelli. And this is also an area that is completely absent of iris. Iris will be expressed all throughout this domain and will remain absent there. So this is going to start off as being a disappointment, because what happened was when you overexpress twin of iris, cynoculus, or doxin, you can clearly see that we just kill the ocelli and transform them into head epidermis. Not at all what we were hoping to see. And of course, one thing that you can kind of note is that, remember, we're turning on the entire left end of this core pathway. And one thing that we knew about going into this is that cynoculus is actually a very interesting molecule in that it forms 2 types of complexes. It forms an activating complex with izapsin and promotes gene activation, but it also binds to the Groucho family of co-repressors and shuts down expression to the same set of genes. And this happens both in flies and in vertebrates. So when we were turning on all these different molecules, we were actually making a mess of that entire tissue by trying to turn on and shut off the genes at the same time. So that result really wasn't giving us much information. So what we wanted to do is to force this cynoculus to behave as this activating complex. And so we did the following set of experiments. We actually went back into our lab collection of reagents we had made over the years, and we decided to create a molecule that, a chimeric molecule fusing cynoculus to the VP16 activation domain. So, but we needed to make sure that we found an activation domain that was just as strong as I's absent. So we did this very similar yeast transcription assay where I've shown you ILIS before. You can see cynoculus by itself doesn't activate transcription at all, really. It's just a DNA binding protein. Its co-activator, I's absent, is really strong, even stronger than ILIS. It can have yeast cells survive even under 250 millimolar concentrations of the inhibitor. So we needed to find an activator that when fused to cynoculus, that would be at least as good as I's absent or better. So we chose VP16 after going through a large number of activation domains. And what you're seeing here is a luciferase assay where you can see our GFP control doesn't activate at all. Cynoculus is pathetic, just like it is here. I's absent, for all its strength, can activate our luciferase assay to a little bit. Here's the SO-IA complex, which of course would be better. And now we have SOVP16, which is dramatically stronger than the cynoculus I's absent complex. Now the question is this going to be toxic or will it be biologically relevant? So in the paper that in which we generated this reagent, we had actually done rescue assays. So we took a cynoculus 1 mutant and we drove expression of this strong activator and could achieve full rescue, not only in quality, but in the number of ommatidia. All of the photoreceptors were in the right place. Nothing was awry. If we fuse cynoculus to the engrailed repressor domain, we fail to rescue. So this molecule is biologically relevant, and it's due to the VP16 activation domain itself. Now, when we express that molecule, then we get this dramatic result where we can actually get the 3 ocelli to now turn into compound eyes over here. So now we have a 4-eyed fly instead of the 2 laterals. And you can see in the standing EM a little bit better than this light micrograph. You can see the individual ommatidia, you can see the lenses that are formed, you can see the little mechanosensory bristles that are normally present in the compound eyes. If we take sections through those, you can see, I've marked them all, you can see where are we here, the pseudocones, you can see the lenses, which you saw now in section. You can actually see little clusters of photoreceptors. There should be an inset on this slide, and you can begin to see all the open rhabdome, and you can see individual rhabdomeres. Now, there are very few ommatidia in these dorsal eyes that look completely normal. So we have not gotten a beautiful compound eye with nice 7 or 8 photoreceptors, but it has done pretty well. We're actually collaborating with Robin Hiesinger, who's a friend of Arash and I as well, to actually trace the path of the axons, because as I mentioned earlier, the normal ocellar axons actually go straight from the ocelli down to the lobular plate, whereas the compound eye ones will go through the lamina, the medulla. So if this transformation has gone all the way to the end, we should now redirect the ocellar axons to the lamina and the medulla as well. We're actually collaborating also with Ori Schaefer and Maria Fernandez, a couple of friends of mine, that are looking at circadian rhythms and sleep, because the eyes seem to play a major role in those 2 behaviors. So how do these 4-eyed flies actually compare to eyes that normally have 2 compound eyes and ocelli? I wanted to quickly show you that the development of these things, at least in the larval disc, approximates what happens in wild type. So we've stained them with a number of different markers. You can see here that you have iris turning on in this band. So here's the normal expression of iris ahead of the morphogenic furrow. Here you have a second band of iris. You have doxin coming on in all the different photoreceptors. Here's I's absent, which we had forcibly expressed. Both hedgehog and DPP signaling are important for moving the furrow across. So if you look at the terminal transcription factor of hedgehog pathway, CI, or the phosphorylated version of MAD, which is the terminal part of TGF beta signaling, in both cases, there's a band where you would see the normal morphogenic furrow. You can now see a second band of expression showing you that there's a second furrow coming in from that dorsal head vertex. You can see now that you have clusters of ommatidia here. You're now getting clusters of ommatidia within the new transformed dyes. And we've looked at a bunch of other markers as well, and all of them show up just like they would in the compound dyes. So our kind of model was this, that based on what we're thinking, that there's some stoichiometric balance between these repressive and activating complexes. They don't have to be equal, but they have to be some balance that will give you ocellular development. If you shifted the balance towards a more repressive complex, you might kill the ocelli. If you shift it towards these activating complex, you would transform them into those tissues. So in the next slide, we're going to manipulate both izapsin and Groucho and actually show you that that is indeed the case. So over here at the end, you see me overexpress Cynoculus and you kill the 3 ocelli. If you overexpress Groucho itself, and you basically flood the system with Groucho, so it takes all of the Cynoculus that's normally expressed and makes it a repressive complex, you will actually kill the ocelli too. If you knock out izapsin, basically depleting the ocelli of izapsin and forcing a repressive complex, you will again kill the 3 ocelli. So these are all consistent with each other. So the other 3 are now consistent with each other in the sense that if you overexpress that activating version, SOBP16, I've shown you get the transformation. If you now forcibly express the activator izapsin, you basically flood the system, grab all of the Cynoculus and turn it into an activating complex. For the most part, you can actually force it. And here in these Groucho lung mutants, which I think this part is hilarious because this mutant has existed since the 1930s, and no one has really ever noticed that that field actually goes from those 3 ocelli to this large field that becomes faceted if you look really closely. And so in this case, we're depleting with a genetic mutant of Groucho, and you're now shifting it to this izapsin-like complex and shifting its fate. We know this is Cynoculus dependent and not dependent upon anything else, because if we take a Cynoculus 1 mutant, which you've seen before, you now express izapsin in wild type, and now you combine the 2 things together, you can see that without Cynoculus, you cannot rescue the compound dyes, nor can you transform those into ocelli into compound dyes. So I wanted to revisit 2 of these experiments. So in the past, I had shown you that if you overexpress twinovirus and Cynoculus, initially that led to a loss of the ocelli. If we do those same experiments, but now get rid of Groucho, so when you activate twinovirus, you're turning on Cynoculus, but you're now forcing it to be an activating complex, or you just put in more Cynoculus here, you can now see in both this case and that case, you can start to see the transformation back to compound dyes. Now, hopefully, well, maybe not hopefully, but at this point, you're probably thinking, well, he told us a long time ago that you can just generate ectopic eyes when you overexpress these retinal determination network members. So could this simply be that you're generating an ectopic eye, and in the process of doing that, you have killed the ocelli, and that it's not really a transformation? So and people have asked me this over the last few months I've been talking about this. So I'm going to belabor the point a little bit with a few slides to kind of hopefully convince you that these are real transformations. So the first thing is sort of just, it's not real proof, but it's more anecdotal information. There's a mutant that Jim Price had identified many years ago, and also Bill Stark had identified it before him. And it's called extra eye. And that extra eye mutant has, as its namesake, an extra compound eye within that dorsal head vertex. But what you can kind of see is that when you have that extra eye, you still have your 1, 2, 3 ocelli. So those 2 structures can inhabit the same physical space. You don't have to lose one at the expense of gaining the other. But that really isn't proof. So what we did is we took advantage of the ability to control the expression levels of our RNAi lines. So Mark Potashny had shown that GAL4 in yeast is temperature dependent. The higher you raise the temperature, the more activity GAL4 or the better GAL4 will work. Andrea Brand, when they developed the US GAL4 system for flies, had shown the very same thing. So you can grow flies at different temperatures, and you can actually increase the amount of your transgene that you're expressing. So we took advantage of that fact by comparing a loss of function, where we knock out and kill the ocelli, wild type that doesn't have any transgenes in it. And now on top of the wild type, we add the transgenes and we grow flies at either 18 degrees, 20 degrees, 25, and ultimately 30. And what you can kind of see is sort of an evolution of that transformation. So at 18 degrees, you begin to see multiple ocelli starting to form, and they make this little ring. You can see that they're sort of connected. By the time you get to 20 degrees, just 2 degrees higher, you can see that all congeals into one mass and it's beginning to look faceted. At 25 degrees, there's more facets. You now begin to see the compound eye pigment colors are coming in. And then finally, when you shift it to 30, you can see this transformation to compound eyes. So we're able to see that structure is changing from one visual organ to another, just as you raise eyes absent levels. And then I hope this last proof is going to sort of help. So remember I showed you the OTD1 mutant kills the 3 ocelli. So OTD is this early transcription factor that's expressed in that ocellular domain. You actually needed to specify the ocelli. So if this was not a conversion, if you were simply in generating an ectopic eye, you should be able to knock out OTD and then still transform the head epidermis into compound eyes, just an ectopic eye induction. So what we did is, though, we combined that OTD mutant with our ability to express eyes absent, and you actually do not get the conversion. So you actually have to make ocelli first with OTD, and then raising the levels of the RD network will switch those ocelli to compound eyes. The flip side of this argument should work as well. We've been showing you how we can raise levels of this network in one tissue and switch it to another. We should be able to drop expression in the compound eyes and get the reverse transformation. So Claude, who I'll show you his picture at the end, the grad student who's been doing all this work, he spent a lot of time getting the right GAL4 drivers, RNAi lines, mutant combinations together. He found one that actually will work. And what he found was if you combine one of the iris alleles that gave us that really low level of expression, maybe about 2 or 3%, and he combined it with an RNAi line that's driven by an eyes absent GAL4, so tempers twin of iris levels at just the right point, you get these dramatic transformations. So the compound eyes are missing, and you can transform them into a maxillary palp, which you can see here. They get transformed into 2 antennas. Here's the normal antenna on that side of the head. You can actually get just head epidermis, and hopefully, if you can see carefully in orange, you can see there's a nice ocellus forming in its place. The normal 3 ocelli, here's one, and the other 2 out of focus, but here's one that's developing instead of where the compound eye is. So we can toggle back and forth between these 2 systems and actually get this reverse set of transformations. So the idea overall is that in different tissues, you have different levels of PAX6 using different combinations of these factors, and they give you totally different outputs. So if you use both members, the stronger and the weaker, to activate the same downstream targets, you're going to make a compound eye. If you get rid of the strong one and only use the weak, you're actually going to get ocellular development. If you kill these 2 things together, all of this network is actually going to collapse. It actually drops down to very low levels because there are other additional inputs that come in outside of PAX6. You can actually convert those compound eyes into head epidermis. And I've also shown you can turn it into ocelli, maxillary palps, and antennas. Now, this model, I think, is actually, it's kind of helpful because there is lots of transcription factor families. There's a Drosophila gene cripple. In vertebrates, there's about 17 members of these, and they all bind to the same target sequence. they're expressed in many overlapping patterns, but their expression patterns can also be mutually exclusive. And so by raising the level of, or lowering it or calibrating it to some point, you can get a myriad of different outputs using the same network, which I think is a kind of a nice way, a nice way to augment the way we think about gene regulation. And then the most important slide, of course, is to show you that all the work was done by a wonderful graduate student, Claude, who is actually graduating this year, and actually in a couple of months, and will be moving to Melanie Worley's lab at the University of Virginia. She was a Ishwar Hariharan postdoc, and she and Claude are going to look at regeneration and single cell work in the wing disc. And all of this was done at Indiana University. I do want to make one more plug for a student in my lab. So I didn't talk about Haley Brown's work because she was just here a couple of weeks ago interviewing with Melissa Harrison. She's going to join her lab as a postdoc. So she has an equally beautiful story on how the compound eye and the developing wing balance their fates during development using polycomb repression. So Haley should be here August 1st. So, you know, please welcome her and enjoy her. She's a fantastic student, and I think you'll enjoy interacting with her. With that, thank you very much. And if there are any questions, I'll be happy to answer them.
Development, the order of the eyes, what would the significance of the ocelli developing after the compound eyes be if the thought is that ocelli evolutionarily developed first? Does that make sense?
Right. So, so in the order of the fly, why do the ocelli come up later? You know, I don't really know the answer to that question. It's a really great question. What I can tell you is that late development, even when we look at genes that are responsible for cellular development and we try to figure out when their mode of action is, it is always after those same genes are specified in the compound eyes. But I don't know why it would develop later in time, even though it was the sort of evolutionary ancestor. But that's a great way to think about it. I just don't know. Yeah, David.
Sort of a trivial question, but I noticed that when you transform the ocelli into compound eyes,
Yes,
you only ever got 2, you never got 3.
yes,
Or at least you didn't show 3. Is
right.
there something about that? The 1.5 really bothered me. How 2 1.5s can make 3? Where's that third one?
Sure. So it turns out that this Russian scientist, his last name is Zarkolov. And this guy in Birmingham, an English scientist, independently showed in the 1940s, what they did is they would cut open the larva and they would pull out one eye antennal disc and let the animal heal. And when it came out, when it died as a ferret adult, you would get half a head and it would make one lateral ocellus and the medial ocellus look like a split pea. And so that's how they knew that each one was giving you 1.5 of the 3 ocelli. And so during pupil development, these 2 eye antennal discs will actually surround the brain and they will fuse together and they'll stitch themselves up. And those 2 split peas come together to form one perfectly round ocellus. Now, the reason why we only get 2 out of the 3 is because that DBE Gauss-4 driver that we use encompasses the entire zone within each disc. So that 1.5, actually, that whole area becomes one compound eye, the 2 things merge. We do have Gauss-4 drivers that are expressed in only one of the 2 types of ocelli. So we have a Gauss-4 driver that's only on the lobes. The reason why I don't show those, they do turn into compound eyes, but the reason why I don't show them is because the compound eyes have like 4 ommatidia. Because they're so small, the expression patterns are so tiny, they don't make dramatic pictures for me to show you. You have to kind of squint, look at it. Whereas this, you just get these massive eyes. But we can get individual ocelli to transform. Great question.
Very interesting. And when you ectopically express a toy, you can generate the ocelli cells, right?
Well, you know, that's the funny thing. So when we and others were first discovering we can get it topic-wise. We would all see cases where there were like these small pigment patches and with different Gafford lines, and we threw those all away because we were looking for robust compound dyes. But I do think that what we were seeing in some cases were Gafford drivers that were turning on were expressed at really low levels, and those little pigment patches may have been ocelli. So I asked Claude to actually do some of those experiments, and he just kind of laughed at me because he wanted to graduate and said, "There's no way I'm gonna do this." But I'm gonna do them myself this summer because I have all my notes, and I think we can find twin ovitis generating little ocelli.
I see.
Yeah,
So that's a related question. So when you knock down the iris,
Mhm.
the ocelli, I mean, the toy is still expressed in that area, but
Yes.
its eye is gone. You don't see the ocelli in that area, is it?
No, the only time you'll see the ocelli is if you knock down both genes to some medium level, so. Yeah, that is sort of
So
one
is
whole-
that is toy expressed in that area in the compound eye
Yes,
is not good enough to.
it's not enough to make an ocellus. It actually, so in the eyeless mutants, even though some animals give you no eyes, that's actually a minor fraction. Most animals give you that small compound eye. So we think that levels of twin eyes are just too high and you get a compound eye development. And then when we lower them together, that's when we get the ocelli.
That's why you have to kind of modulate them.
Exactly, yeah,
Thank you.
sure.
Think that, so in terms of trimavirus and do they bind to the same? Because there's also these new domains that can sort of interact differently and you can bind to different sites, different affinities, even though they're really similar. Do you think they're binding to the same regions or are they also binding to different?
So my preliminary look at cut and run suggests that they bind to mainly the same regions, but there are places that they bind individually. More than I thought they would. When I looked at, so when I associated their peats with their genes, the areas that are, most of the areas that are unique to one or the other, those genes are not expressed in the eye itself. But I will say that even some of the places that are expressed together, some of those genes aren't in the eye either. So I do, and I think the home domains, I think, if I remember last I checked, were like 95% at the amino acid identity similar. So and within the recognition helixes, they're not identical though. That's the thing, so.
I mean, I was just wondering, because like, right, with the intrinsically disordered gene activation, there's interesting evidence that those are actually not just about interacting and activating, but also changing their association with DNA. To some degree, so that's what I was like interested in.
One thing that we could do is with our chimeric molecules
Yeah.
and our deletions, we can now redo our cut and run and ask whether changing those disordered domains potentially.
Yeah, I mean, that could be interesting,
Change
right?
our
Does it look now more like the other one?
Yeah, that's a great idea. We haven't thought of that, but that's really good. We can do that. Absolutely, yeah.
So you mentioned that the strength of activation of gene expression kind of depends on that activation domain, yeah?
Yes.
So this is a very naive question. Is that like a protein intrinsic activation domain, or is there something that regulates that, like another protein that you can also maybe target to mimic the strength of?
So both iris and trilovirus, their activation domains physically interact with subunits of the mediator complex. And then that has an indirect effect on releasing RNA Pol2. But I think they interact with the same subunits. So we could try to manipulate the subunits of the mediator and see whether that could change it.
What makes it strong versus not so strong, I guess?
We don't know that. That's one area that's been out of my area, so I have not really investigated, but it's a really good question. Yeah, I don't know what makes it stronger, to be honest. When you look at the series, the repeat size, that proline serine threonine repeat, the iris one is much longer than that twin of iris. But I don't know how that actually relates to stronger versus weak. Yeah, but it's a really good question.
Actually, if I can follow up with it,
Sure.
and indulge the audience for a minute longer. this quantitative or stoichiometric model that you presented, how can you use it to explain mammalian eye development, right? Where eyeless is showing loss of the iris, so it's an iridia.
Right.
Whereas in flies, it's the entire eye field, but not in mouse. Are there networks stoichiometrically acting differently like this in mammalian eye development?
Well, I can tell you for eyeless specifically, so eyeless, there's only one copy of PAC6 in invertebrates. And it is expressed at dramatically different doses within different tissues in the developing eye, in the developing brain, in the pituitary. And there are some instances where heterozygotes will cause phenotypes in some cell types, but not others. And so I think it's levels, in that case, you're basically calibrating the levels of a single transcription factor differentially. In the case of eyeless and twin of eyeless, you keep their expression levels pretty much the same, but you're modulating the strengths of their activation domain. So it's just a slightly different way of getting to the final product.
Well, thank you very much.